Hi everyone, today we're going to be talking about polymerase chain reaction, or most commonly known as PCR. Now you may have heard of PCR because of COVID-19. We're going to be talking about the basic PCR steps or how PCR, the basic structure looks like. Alright. Once the sequence of a DNA fragment is known, a technique called the polymerase chain reaction, or PCR, can be used to make millions of copies of a specific region of a DNA fragment. So we use PCR to make millions of copies of a specific region of a DNA fragment. So we're amplifying a certain region of a DNA fragment. Now, PCR is extremely sensitive and it can detect a single DNA molecule in a sample. PCR is useful because this single DNA molecule can then be copied or amplified. So to make millions of copies of it means we're amplifying the DNA, so we're making more of that DNA fragment. So we're copying it, right? We're putting it into a copy machine. That is called amplifying. Numerous times to be used for DNA analysis. So once we have many copies of this certain DNA fragment, we can then do DNA analysis. So use gel electrophoresis or find the sequence of this specific DNA or study this DNA or look at this gene. But we first have to amplify. We have to have more of this DNA fragment. All right, let's look at the steps of PCR. The steps of PCR are divided into three main steps. The first one is denaturation. The second one is annealing. And the third one is extension. Those are the three steps, or the three main steps for PCR. So in the first step, called denaturation, we are separating the DNA strands. What does that mean? Let's look at this diagram right here for step one, which is denaturation. PCR is performed by placing the DNA fragment to be copied with the DNA polymerase and the 4 DNA nucleotides and 2 short single-stranded pieces of DNA called primers in a tube. So the first thing we gotta do is add our DNA fragment that has our gene of interest, our DNA polymerase, the 4 nucleotides, and 2 short single-stranded pieces called primers in a tube. Once we have them in our tube, in our little Eppendorf, the primers are complementary to the ends of the DNA fragment that will be copied. So this primer is complementary to the DNA fragment that will be copied. So let's say, for example, I have this really long fragment and I just want to copy from around here to here. So the primers that I have will have complementary bases to the start of this area. So why do we need primers? Primers are basically the starting points. They are the starting points for DNA synthesis, because if we look at DNA replication, DNA polymerase cannot start replicating on its own. It needs a starting point, and that starting point is our primer. Now, let's look at a couple of things before we talk about the process. So obviously we need DNA polymerase so that we can add the nucleotides and things like that. The thing that I want to mention is that we have heat-resistant DNA polymerase, and that polymerase is called Taq polymerase, and we find Taq polymerase in hot springs, and it is in the bacteria that lives in these hot springs. Now, the reason we use Taq polymerase is because Taq polymerase is thermophilic, or it's heat-loving. Right? It can handle really high temperatures, which is very important because PCR is based on very, very high temperatures. So for the first step, which is denaturation, if you talk about enzymes, or if you remember enzymes, we know that denaturation is basically a rise in temperature that would kill these enzymes. Well, if our DNA polymerase cannot withstand such high temperatures, well, then we can't use it in PCR. So that's the thing that we've got to notice, is that we have a heat-resistant DNA polymerase, or Taq polymerase. Okay, now, first step we said was denaturation. What entails in denaturation? Well, step 1 is basically the DNA strands are separated by heating. So we heat up the tube to up to 94 to 98 degrees Celsius. Okay, and this will cause the hydrogen bonds that are holding these two strands. So we know that the nucleotides here are connected by hydrogen bonds. So we have these two nucleotides, and then we have the hydrogen bonds connecting these two nucleotides together, and that's what's connecting these strands. So what happens when I raise up the heat, I'm actually getting rid of those hydrogen bonds, and therefore I'm separating the DNA. So we know that our DNA is double helix. In denaturation, which is in step 1, the DNA strands are separated by heating. Right, now let's look at step 2. So once the heat has separated the two strands of the template DNA fragment, the tube is then cooled. And we cool the tube to around, it ranges from maybe 45 or 55 Celsius to around 72 degrees Celsius. Okay, depends on what we're doing. So we cool down the temperatures, and the reason we do that is because, as we said, step 2 is annealing. The reason we do that is because we want the primers to attach to those single-stranded DNA. Again, in the first step, we've got denaturation. We separate that DNA double helix using heat. And then in step 2, we cool down the temperatures so that our primer can now bind on the single strand of the template DNA. An automated machine called a thermocycler is used to cycle the tube containing all of the components involved in PCR through various hot and cool temperatures. So this thermocycler is what's actually raising and cooling the temperatures depending on which step we're in. Alright, so we have our primers attached to our single-stranded DNA. What's next? Well, the next step, or the third and final step, is called extension. And obviously from its name, that means we're extending something. What does that entail? Well, that means that our DNA polymerase will latch on to our primer and we'll start adding nucleotides to the, to the strand, right? So as shown in the figure, each primer is made to bind to one strand of the DNA fragment. Once the primers are bound, DNA polymerase incorporates the right nucleotides between the two primers, as in DNA replication. This process of heating and cooling and nucleotides incorporation is repeated 20 to 40 times, resulting in millions of copies of the original fragment. So what we see here in step 3 is that, or in extension, the DNA polymerase extends the complementary strands by adding specific nucleotides. So the temperature in extension is around 72 degrees. So what happens is this DNA polymerase will latch on to our primer and we'll start adding our nucleotides until it finishes that strand. Okay, so once we're completed, now we have two double-stranded DNA. So once step 3 is completed, we have two identical copies of the target DNA resulting from the first temperature cycle. So a cycle is going through those 3 steps, right? And once we go through those 3 steps once, we get 2 of the template DNA. So we get 2 clones or 2 copies of that template DNA. If we repeat the process again, This DNA will make 2 copies of DNA, and the other one will make 2 copies of DNA as well, and you'll end up with 1, 2, 3, 4, 5, 6. Sorry, you'll end up with 4. You'll end up with 4 newly made template strands. So you'll end up with 4 copies of that strand. And if you do that again, you'll end up with, this will make 2, this will make 2, this will make 2, and this will make 2. So you'll end up with 1, 2, 3, 4, 5, 6, 7, 8. And then the process just keeps going on and on and on. And once you go through 20 to 40 cycle, now you've got millions of copies of that DNA strand, that original fragment. Now, because the separation of DNA requires heat, the DNA polymerase, as I said before, used in PCR, should be able to withstand those high temperatures, and we said that it's stacked polymerase. And you can find it in hot springs, like the one used in, like the ones in Yellowstone National Park. Now, because PCR can detect a single DNA molecule in a sample, it has become one of the most powerful tools used by scientists. PCR is not used only by researchers in laboratories, but also by forensic scientists to identify suspects and victims in a crime investigation. Obviously, when you find a drop of blood that contains DNA, you're going to amplify that DNA, make more of that DNA so that you can further test or do some DNA analysis on it. Okay? And it's used in crime investigations. It's also used by doctors to detect infectious diseases such as AIDS and, as we saw, like COVID-19. Now, the process used in COVID-19 is a different type of PCR, but these are the 3 main steps of PCR, the simple PCR steps. Now, just to summarize this part of the lesson about genetic engineering, let's talk about the different processes that we talked about, their function and their application. We know restriction enzymes, and an example of a restriction enzyme is EcoR1. Its function is to cut or cleave DNA strands into fragments. Where do we use this? It's used to create DNA fragments with sticky or blunt ends that can join with other DNA fragments. And as we said, it's very important to note that both the sticky-ended or the blunt-ended DNA can join together with other DNA fragments. We talked about gel electrophoresis, and we said that it separates DNA fragments by size, and it's used to study DNA fragments of various sizes. So it helps us look at the size of the DNA strand that we're working with. We talked about recombinant DNA technology, and we said that it combines, that's our key word, combines DNA fragments with DNA from another source, and that DNA from another source is called exogenous DNA. Applications, it's used to create recombinant DNA to be used to study individual genes and genetically engineered organisms and in treatment of certain diseases. So it's used to study individual genes, to genetically engineer organisms, and in the treatment of certain diseases. We talked about gene cloning, and we said that it produces large numbers of identical recombinant DNA molecules. And it is used to create large amounts of recombinant DNA. Why do we need those large amounts of recombinant DNA? To be used in genetically engineered organisms. We talked about DNA sequencing, and we said that DNA sequencing identifies the DNA sequence of cloned recombinant DNA molecules so that we can further study that fragment. Its applications, it's used to identify errors in the DNA sequence, and by errors we mean mutations. We use it to predict the function of a particular gene, so that's number 2. And we use it to compare other genes, other genes with similar sequences from different organisms, and that's number 3. The last thing we talked about was PCR, the polymerase chain reaction. And we said that PCR makes copies of a specific region of sequenced DNA. So you've got to really make a difference between PCR and gene cloning. Gene cloning is to make large numbers of identical recombinant DNA, whereas PCR makes copies of a specific region in a DNA fragment. It is used to make copies of DNA for any scientific investigation, including forensic analysis and medical testing. And that would be it for the genetic engineering and genetic tools that we talked about for this lesson.